Hello dear colleagues, today is the end of July 2017 and I will present you the recent case study in determination of causes of geothermal well clogging or plugging, case study from Southeast Europe and only microbial aspect. So let's start. Okay, a little bit about author of this presentation. I finished bachelor, master and PhD at University of Belgrade, Earth Science with specialization in hydrogeology environment. Currently I'm at specialization at Johns Hopkins University, environmental engineering, over 25 published papers, and I have wrote 2 books about groundwater modeling and contaminant transport. Just a little bit of marketing. This is my website and it's advanced groundwater technologies. Just type it in Google, you will find it. We have YouTube channel which is where this recording will be published, LinkedIn, and yeah, take a look at it. Thank you. That's about introduction. Let's go to okay, first a general story. What caused the clogging of the wells? Physical, we have physical, chemical and biological clogging of the wells. This is typical bridging, where particles, smaller particles are coming, and if they come in the same time, the bridging form, like this, where I'm moving my mouse, or this, and this is a good place where bacteria will attach and continue to bind these particles. Okay, let's switch to, I will not speak about chemical causes of clogging of water wells. This is just biofouling of water wells. After the most recent investigations, hydraulic of wells, American Society for Civil Engineers 2014, 80% causes of clogging or plugging of water wells are biological origin. Okay, this is the professor who influenced me a lot. I don't know, he published the first book which I read in 1997. This is, I think, from 2008 or 9, I'm not sure, second edition. Warmly recommend to read it. That's where I start my investigation 10 or 15 years ago about biofouling. Okay, here is the culprit. Bacteria pictured by a 3D microscope. Just a little bit about bacteria morphology: spherical shape, rod shape, spiral shape. Sometimes, especially in the past, people thought that there is no bacteria in groundwater. Okay, this is from Professor Roy Collymore book. You can see that there is bacteria even on 10,000 meters. So I think we solved this question, very often asked by hydrogeologists. This is what is happening. So bacteria attach to surface, which can be, for example, well casing, make a colony, biofilm, and then a complex maturing biofilm. This doesn't look nice, it looked a little bit ugly, but that's actually what is going on, how the biofouling starts, how it develops, and what is the end. So we need to prevent this end picture. I picked this up from internet. This was really good picture. So this is unfold well screen and this is fold well screen. So that's the cause of incrustation and biofouling. Okay, let's talk a little bit about iron-related bacteria. It can be defined as a group of bacteria that utilize iron in their metabolic functioning. It was discovered in 1836. What is not known by a lot of people is that iron bacteria emerged in Berlin, where the whole water main system had to be replaced because it was clogged with iron bacteria, 1877. Okay, this is even more important. I'm now in Southeast Europe, but Cullimore and McCain 1974 carry out survey of 150 countries related to groundwater bacteria. Results show that iron bacteria occur on all continents except Antarctica. Oops, sorry for this. I need to quit my Skype. Show that iron bacteria occur on except Antarctica, for which no data is available. Okay, this is again from the book. So we have red, we have black, gray, and white regions. White regions means that bacteria is not detected, but they are not sampled at all. So it doesn't mean that white area show us that there is no iron bacteria. Basically, iron bacteria has three groups: ribbon formers, tube formers, and different heterotrophic bacteria. Ribbon formers, I will just in a second I will show you the picture. It produces a super long tail which is discarded, and then the cell can swim off through the water to start all over again. Okay, this is very nice pictures. So this is the ribbon. It's detached and swim in the water to attach to another place. Tube formers. This is interesting. This is where incrustation on bacteria is growing. This green cycle. Factors that influence iron-related bacteria: dissolved iron, pH, oxygen, temperature, carbon. I'm just quickly passing through this because this can be known by most of you. Just want to make introduction to geothermal well biofouling, which is very interesting because it's geothermal. We have direct evidence of bacterial event, visible manifestation, slimes, the coloration, cloudiness, coloration of water. So odorous signal: rotten egg smell, fish smell, earthy smell, fecal, fresh vegetable smell, chemistry smell. And how we detect? We detect on different ways. Very popular ways are BART biodetectors. Major advance of biodetectors is the recognition that, as in biofilm, the water contains complex consortia of bacteria. So we don't want to spend a lot of time detecting one bacteria. We want to see consortium of bacteria. This is how BARTs are looking. It's a bottle, small bottles. At the bottom you have nutrient media for bacteria growth. We have a ball, floating device, which restricts the oxygen diffusion in a ball, in a tube. So here is the area where anaerobic bacteria is growing, and here is the area where aerobic bacteria is growing. Okay, this was the picture from 2009. That was the first time I ordered the BART biodetectors. It just came, you see, in DHL. Okay, let's now go to case study. This is case study in Southeast Europe, and this is geothermal well. Its depth is 1,100 meters. It's geothermal well, and it's used for greenhouse heating. And as you see, it's a flowing well. So what was the problem? Actually, the problem was that the pump needed to be replaced very often. So we needed to examine what is going on. Okay, this is another. So the question was, okay, this is incrustation. Is it a chemical or biological? Let's start with biological tests. This is another. You see biofilm. You have it's a layered, so you have one color at the top, and we scratch it. You have another color, black at the bottom, probably sulfate reducing bacteria. Okay, we take wellhead off, and you see flowing well. These are BART detectors. Young scientists took samples. Okay, why is this incubator? Very simple. It's a geothermal well, so we need to maintain approximate temperature in water well. So that was why was this incubator was used. Very simple one, but it's more than enough. It doesn't need to be in 1 Celsius degree. Few Celsius degree, it's more than enough. Okay, these are parts inside, and now results are coming. First day after the samples were taken. So we take sample for sulfate-reducing bacteria, heterotrophic aerobic bacteria, iron-related bacteria, slime-forming bacteria. This is from the top of the geothermal well. You can see that even after 1 day, iron bacteria was shown. You see here it's very aggressive. It's a red color. Okay, I want to show you this. This is from deeps, from depth of geothermal well. And this is methylene blue. It's a replacement for bacteria for oxygen. And you see the bacteria did not take the oxygen at some depth. But at the bottom, you see it's bleached out. So that is the confirmation that they exist. Okay, second day. Again, yeah, we know that we already know that iron bacteria is present. You see the color, this green color. Heterotrophic bacteria, aerobic bacteria is bleached out. Slime bacteria start to show the signs, and sulfate-reducing bacteria is not showing signs. They usually show signs a little bit later, after a few days. Third day again, even worse again. So we don't need to. Actually, we will monitor this. This testing is ongoing. And actually we will monitor for 10 days because of sulfate-reducing bacteria. They can show later, but it was shown that even after 1 day, a big population, large population of bacteria, different kind, is present in geothermal well. Okay, what are conclusions and recommendations? The cause of incrustation of the pump of geothermal well is bacteria. Even the temperature in the well is actually 56 Celsius. Samples were taken to not just determine that, but as a reference before the well rehabilitation. So I will record another video in 2 or 3 weeks, and let's see what is going on after rehabilitation. Did we influence the bacterial aggressivity to lower level, which proves the effectiveness of well rehabilitation? I know that this was very quick, but if you have any question, here is my email. I will be happy to try to answer you. If you have any question, not just question, I mean as I shown you from a book, bacteria, especially iron bacteria, are ubiquitous in the world. And this was the quick presentation. Thank you for your attention. You can find me on LinkedIn, just type my name. And again, if you are interested in what we are also, the other thing what we are doing, please visit our website. So yeah, that's it. Thank you very much, and hope to make another video about post-regeneration, well rehabilitation, bacteria detection. Have a nice day. Bye.